Okay, hello everyone, and welcome to part 2B of sugars and nucleic acids. And today our focus is going to be about the biological enzymatic synthesis of oligonucleotides. So how our enzymes do this in our body. So we are sort of the part 2B, biological enzymatic synthesis of oligonucleotides. And like we did with peptides, we're going to first do the biological, and then the next part will involve the chemical synthesis of oligonucleotides. Okay, so enzymatic synthesis. You've learned this already in some biology classes, and it's pretty intuitive. We have enzymes called RNA polymerases and DNA polymerases, and these make RNA and DNA respectively. And the way that they work is as follows. So I'll draw a strand here, and then imagine this is a DNA polymerase. What we're going to do is we're going to have a short primer, and this primer is needed to, in some ways, act as the initiation site for polymerization. And then all of these deoxynucleotide triphosphates will come in, and you'll get polymerization of the complement strand, and it will match hydrogen bond complementarity to whatever this strand is. This is a highly efficient process where tens of thousands of units are placed in per second. Okay, so highly efficient. Okay, so at the end of the day, you get now a fully double-stranded oligonucleotide, as I've shown here. Okay, so let's take a little bit of a closer look at what this looks like. And as I mentioned, this is catalyzed by enzymes. So DNA polymerase will make a DNA copy when it needs to replicate. Okay, so you've learned about that already. And RNA polymerase makes this when it needs to make RNA, and this is called transcription. This is also known as the first committed step of gene expression, so DNA to RNA. So as I mentioned before, a primer strand here, this is right from your textbook, and then the nucleotide monomers come in and get filled in. Polymerase really facilitates this reaction, and you can see here that you have a double-stranded helix that is initiated and that's recognized by the polymerase. And then the polymerase really sort of acts like a Pac-Man, and then just quickly goes off of the strand and inserts those nucleotides. If we look more closely, so from like a chemist's point of view, in terms of what is happening from a bond perspective, let's take a look at how polymerase works and how it reacts to make a new DNA strand. Okay, so what I have attached here on the upper left-hand corner are thymidine and guanidine deoxynucleotides. And as you can imagine, this came from the growing chain or the primer. And then DNA polymerase is necessary to initiate that nucleophile to the electrophile, in which we now have a nucleotide triphosphate. In this case, it's a deoxynucleotide triphosphate. And what you can see here is that you have a triphosphate, you have a relatively good leaving group, this diphosphate here, and your electrophilic center is actually going to be this alpha phosphate. So that's alpha, beta, gamma. So this is your electrophile. And this hydroxyl group is your nucleophile. Okay, so the polymerase orients this in an efficient position, and to make the leaving group even better, to avoid a polyanionic charge, Magnesium 2 plus is used to help stabilize that leaving group. So it coordinates between both of those oxygens. So polymerases need magnesium to make this reaction go efficiently. Magnesium stabilizes the leaving group. Okay. So these come into close proximity into one another. So the polymerase orients them in the right orientation, brings them close by. And then what happens now is that there's a secondary magnesium that comes in, and this will coordinate with both of these oxygens here. Okay. Inherently, what this does is this stabilizes the nucleophile, makes it a little more nucleophilic, and makes this phosphorus more electrophilic. You can see just adjacent here, you have an oxygen with a positive charge. That oxonium ion really makes this phosphorus much more electrophilic. So when this attacks, you break that pi bond, then the electrons will come back down, and then you'll release and kick off that diphosphate with a magnesium ion attached, which is shown here. So that is your good leaving group. So this magnesium here stabilizes the nucleophile and makes the phosphorus more electrophilic. Okay, so once that nucleophile reacts with the electrophile, you now have your new bond, your new oxygen phosphorus bond. And then to complete another round, this 3' hydroxyl group can now attack another triphosphate. And the process repeats itself and goes on in a very, very fast and enzymatic process. So you can see the direction of synthesis on the growing strand actually goes from the 3' to the 5' direction. So 3' to 5' direction of synthesis by polymerase. Okay, so and here's another drawing here showing the oligonucleotide, in this case DNA. So at each time, this is your new bond that's being made. Okay, so that is some insight in terms of how that works. Couple things I just want to mention too is that this corresponds to DNA polymerase. For RNA polymerase, those are specific, of course, for ribonucleotide triphosphates. So you would have a hydroxyl group here, hydroxyl group here. The polymerases between DNA and RNA are very specific for the monomers, so that you don't get mixed RNA DNA sequences. And a lot of this has to do, of course, with specificity, but also, as we'll talk about later, the 5-membered ribose or deoxyribose ring, they can actually have different sugar conformations, different sugar 3-dimensional structures. And so this is a governing feature that allows it to fit and accommodate into the polymerase active site, so you don't get those mixed sequences. So it's not so simple as to say that you have a hydroxyl group here or you don't, and that's the only change. We get actually very different 5-membered ring structures. Okay, so a little word about triphosphates. So triphosphates are inherently very reactive, and as I mentioned, you have an alpha, beta, and gamma site, and these can be stabilized by magnesium to make this a better leaving group. But inherently, you can get attack occurring at alpha, beta, or gamma, and it really depends on what is being made. In the case of transcription or replication by polymerases, we would see attack occurring at the alpha phosphate. But if triphosphates are being used for things like phosphorylation in order to install like a phosphate on a hydroxyl group of serine, for example, or involved in things like glucose metabolism, as you've learned with metabolism, then oftentimes you'll get the gamma phosphate being attacked, okay? So glucose metabolism or enzyme regulation, and really how do you know how this is going to work? Well, this really depends on how the enzyme is set up in order to orient the nucleophile with the electrophile. Now, it turns out that these triphosphates, they are electrophilic, but you really need some type of magnesium to help make that a better electrophile to stabilize that leaving group. And they are also, they're not super reactive either. So, yes, triphosphates will not last a super long time in solution, but you don't really have to worry too much about cross-reactivity in the absence of enzyme. So it's really a delicate role in which the enzyme can bring them together, and then you catalyze that reaction. Nature faces some problems inherently as well. If you have something like an RNA polymerase, an RNA has 2' hydroxyls, the nucleophilic attack during transcription is always on the 3' hydroxyl. You never get mixed sequences at the 2' hydroxyl. Specific attack on that alpha phosphate is required, as I mentioned. And without enzymes, if you have a mixture of NTPs, you would perhaps get a non-specific sequence and the reaction rate is slow. So nature is actually slow in the absence of enzyme, which is good because you don't want to just start getting random sequences, non-specific 2', 3' hydroxyl binding. And then once a polymerase comes into play, which is this Pac-Man looking structure, it's highly efficient and highly specific. Okay, so as I mentioned before, this is a picture of the RNA polymerase wrapping itself around the template strand, and then these ribonucleotide triphosphates coming in, and then it just elongating along the strand. Right, here's a transcription bubble showing DNA on the coding strand, and then the template strand, the RNA strand being made by RNA polymerase, and the direction of polymerization is in the 3' to 5' direction. So it's analogous to replication with DNA polymerases. Okay, so the process to synthesize oligonucleotides enzymatically. So if you were to go in the lab and you want to make DNA or RNA using a polymerase, let's outline what the ingredients are. So number one, you need a template strand, right? The polymerase uses the code off the template strand in order to determine what the complement is going to be. Second, you obviously need an enzyme. In this case, RNA polymerase or DNA polymerase, depending on which oligonucleotide you're trying to make. To initiate this reaction, you need a primer, a short primer. You need obviously ribonucleotide triphosphates or deoxyribonucleoside triphosphates. And then lastly, which is also really important as we've talked about, is the impact that magnesium has. So you need to add magnesium, and often the best way is to add magnesium chloride as a salt. All right, so it seems pretty good and pretty efficient, and it certainly is, but there are inherently some problems and disadvantages in synthesizing oligonucleotides enzymatically. Okay, so I'll just outline a few. There certainly are, there's many many more, but the main ones is that the enzyme polymerase, like in an in vitro solution, they're not perfect. They can make errors. They can, they can lose their efficiency. They can stop prematurely, or sometimes they can go past a termination site. So sometimes you can run the problem of having mixed length sequences and shorter or longer sequences than what you actually desire. Okay, so enzyme can make errors, stop prematurely, or go past the termination site. This process is typically good for hundreds of nucleotides, or even thousands even. So if you have something that's 300, 400, you have an oligo 300, 400 units in length that you want to make, that's great. This will probably work, or even 1,000 in length. But it's very difficult to make short oligomers. You need that primer in place, and then the polymerase, it's a huge enzyme, and literally once it gets started, it really doesn't want to stop. So it's hard for it to actually recognize a template strand that is actually quite short with a short primer. So it's difficult to make short oligonucleotides that are, for example, 50 base pairs or less. And number 3 is kind of related to number 1, but because you can get mixed sequences and you lack that consistency, it can be difficult to purify. Another thing is polymerases, they need to be kept at very stringent cold conditions. So you can't leave the enzyme out on the benchtop, come back the next day. The polymerase will inherently denature and be non-functional. So they do have limited lifespan, which can, of course, make their efficiency not as effective, and therefore you get lack of consistency, and you can get products that can be difficult to purify. Okay, so with that, this concludes our biological enzymatic synthesis of oligonucleotides. Okay, see you next time.